Well, thank you everyone for joining us this afternoon. It's 6 p.m. in Madrid, but I can see that there's people connected from other countries. So doesn't matter what time is it. Thank you, everyone. Today we're going to discuss the new oral microbiome test with Nacho Esteban, the CEO of the company, and Débora Pino, product manager at 24Genetics, in one of the principals developing this new product. She's also joining us from Germany. So thank you, Deborah. And I just want to remind you that we have opened the questions and answers option. So everyone that wants to answer to ask any questions, sorry, just please do not hesitate to use it. We will dedicate some minutes at the end of the presentation for going back to your questions. So anything else on my side? Nacho, you can start.
Okay, hello guys. Thanks for joining. It's amazing to see how many business partners all over the world. And we are pretty excited on showing you this new product that is it's a great breakthrough. As you guys know, our purpose is to help the people on their care of their health and well-being through genetics. And until now, we've been using DNA human tests to show the predispositions of the people and see also the predisposition on drug response and health prevention, nutrigenomics and stuff. But for the first time, we're launching a new product that is not about human DNA, but is something very important for prevention and for the at the end where the health and well-being is going. That is prevention plus precision medicine and personalized treatments. Today we're going to talk about microbiome. Oral microbiome, and this take 24Genetics to be a multi-omic company, not just DNA. That is pretty important for us. At the end, in our purpose to help the people to care about the health and well-being, we have to give the best information we can. That means we have to look for anything that is relevant in order to take the best decisions. So the most relevant information that we can find on a human is its DNA. Is the most important thing we can look at in order to take decision, but it's not the only thing. So this is the first step for the rest of the things out of the DNA. We are starting with oral microbiome. We're starting with oral microbiome because we already have the saliva from thousands of customers all over the world. So it's quite easy to ask for making the first research and development on this product. And it's a real breakthrough on how to treat the dental care. As you can see with this product, we will be able to go to the dentist before we are having a caries or a periodontitis. So till now, the people is going to the dentist just when they are sick, when they have a problem and they have a disease, or they have a caries or periodontitis in order to solve it. Right now, we will be able with this product to go to the dentist before you got the problem. So you can see, looking at your microbiome in your mouth, that maybe in the future you can have a problem of caries, periodontitis, or even oral cancer, and that's a total breakthrough. I mean, this is the right product for the physicians that are caring about our teeth and our mouth in general. And but there is something, because some other companies are able to see microbiome on the mouth for taking care of your mouth, but there is something that nobody has in the market right now, is that we are able to see associations with diseases on our lungs and on our pancreas, and that's nobody have it on the market. So it's not that we are the best or the cheapest, is that we are the only ones, because nobody is able to see this association. So this is a totally different product. It's going to be very useful, and I think it's going to be very easy to sell. So, okay, I leave you guys with Débora, that is going to show us from the very beginning what microbiome is and what is our product.
Hello, everyone. Thank you, Noelia and Nacho, for the introduction, and thank you to all of you for joining us today. Well, as Noelia and Nacho said, I will be presenting this product, and as it's totally different from what we have had until now, I will go into more technical details as well as the scientific evidence that we have based this product on. So let me present my screen. Okay. So, well, oral microbiota. For starting the beginning, what is the microbiota, and what is specifically the oral microbiota? Well, microbiota refers simply to the set of microorganisms that are found in a specific environment or space. Therefore, oral microbiota is the microbiota that we have in our oral cavity. And even though it's less studied than the gut microbiota that we are more used to listen talk about this one, it's anyway the second largest and most diverse microbiota in the human body. It harbors numerous microorganisms, including bacteria, fungi, viruses, and protozoa. We will focus on bacteria. Our analysis studied the bacteria in the oral cavity, and only in bacteria there are more than 700 species of bacteria that we can find in the mouth. We will analyze saliva. In fact, the tube for collecting the sample, it will be the same that we use for genetic testing, the test that we are doing now. But the mouth is much more than the saliva. So just to let you know that there will be a different microbiota depending on what we study. If we study saliva, if we study the throat, if we take the saliva sample, or we take a sample with a swab, that will change. But an advantage that saliva presents is that it draws the microorganisms that are in the surface of the teeth or on the tongue, for example, forming biofilms. This can be also found in the saliva, depending on the concentration that there are in this surface. Then, just as a curiosity, even in the tongue, there are, depending on the part of the tongue that we analyze, there will be different kind of microorganisms depending on their nutritional requirements, as well as the oxygen requirements. So the oral microbiota, well, actually these microorganisms in general are classified depending well, one of the classifications, better said, can be done depending on the interactions that the microorganism that has with the host, in this case with ourselves, with humans. And we will find symbiotics microorganisms. These ones are beneficial for us. It's not only that they won't be harmful and that they are not pathogens, it's that they give us a benefit at the same time that we give a benefit back to these microorganisms. That is a bit more obvious, that we gave them a place to grow and they get nutrients from us, but we also obtain lots of benefits, like they help us with digestions of certain compounds that we won't be able to digest without microorganisms. They also help us with the immune system. They help us to develop a healthy immune system, among much other functions that a healthy microbiota has. Then we have commensal microorganisms that these ones are not harmful, but neither beneficial for us. They are just there, and we don't get benefits or harm from them. And then the pathogens, that these ones have the biochemical structure and the genetic structure to cause a harm in our cells. And we will find the primary pathogens, that these ones are always harmful and they will always cause a damage in the human body. And the opportunistic ones, the opportunistic, they can be perfectly part of a healthy oral microbiota, but in certain situations, such as dysbiosis, that I will explain now in a bit what that means, they will behave as pathogens. Then we classify the microbiota, the oral microbiota, in the core microbiota, that it's composed basically of symbiotics and commensal microorganisms, and this is the same in every human. Everyone has the same oral microbiota. And then we have a variable microbiota that it changes depending on the person, and in some people, it could have a pathogens microorganism. Then, as I said before, there are important concepts that we have to define, that are the eubiosis, and this will be just a normal or balanced microbiota. Here, all the microorganisms are in an equilibrium in a way that they obtain benefits from each other. They also give benefits to us. This will be the healthy microbiota. Then we have a dysbiosis. This is basically an imbalance, a disequilibrium on the microbiota, where the diversity of the microbiota decreases, and there is also an increase of pro-inflammatory species. These pro-inflammatory species, it's worthy to note that they can be also present in an eubiosis state. However, in an eubiosis state, this will be in a really lower the abundance of these organisms will be lower, and there will be also an increase of the anti-inflammatory species, so that's those ones counteract the pro-inflammatory, and we will be in a balance. This said, I want to talk a bit about the method that we use to study the oral microbiota. And we use 16S ribosomal RNA sequencing. The 16S ribosomal RNA, it's a gene that it's exclusively of prokaryota, and it's part of their small subunit of the ribosome. This gene has a particular characteristics that allow us to use it for taxonomic classification, and it's that it's composed of 9 variable regions, but also it has really conserved regions along all prokaryota. And these conserved regions allow us to select this gene, amplify it, and sequence it, and then base it on the target region, that in our case is the B4, we will be able to classify the taxonomy of this bacteria.
Guys, this is quite important, because what we have done now is not that we are doing a microbiome, oral microbiome or microbiota test. Is that we have built the pipeline, all the bioinformatics platform, in order to identify any kind of microbiome. So we have done this part of the bioinformatic analysis with using machine learning software, that we have trained in that software to be able to distinguish and to identify more than 10,000 different bacteria. That means this is our first product, that is oral microbiome. But with this platform, we are able in the future to make a microbiome. So in the following months, we will be announcing microbiomes for the rest of the body. Gut microbiome, that, as you probably know, is the one that is affecting more our health and wellness, and other kind of microbiomes that are going to be very helpful in order to take good decisions. Thank you.
Thank you, Nacho. Yeah, exactly. The pipeline will allow us to identify any bacteria, even if it's common of the oral microbiota or not. So it can be applied to other microbiotas and environments too. Then, as I said, we use this analysis for taxonomic classification. This is a taxonomic classification from phylum or even domain, that will be just saying that it's a bacteria, until a strain, that will be the deeper analysis. With this one, we arrive to a species or genus, but in most cases we will be arriving to a species. So we have a really good precision with our pipeline of analysis at this moment. Then, as I said before, we have a core microbiota and a variable microbiota. So therefore, there are factors that are affecting our oral microbiota, and that are the ones leading to those changes. These factors are both exogenous and endogenous factors. On the exogenous factors, we find diet, alcohol consumption, and smoking as the main factors causing change. And there is also even though it's not in this figure, there is the antibiotics. In fact, we don't recommend to anyone to take this test, and it's important that you have it in mind when you sell it to your clients. We don't recommend taking this test while taking antibiotics, because it can affect, of course, the microbiota, and the results will not really reflect the health status of that person. And then as endogenous factors, we have genetics, ethnicity, and age as the main ones. There is also pregnancy, and the same as with antibiotics. We don't recommend to pregnant women to take this test, because during due to hormones change during pregnancy, the oral microbiome changes a lot, and therefore it won't be reflecting the health. Maybe we could arrive to wrong conclusion, saying that it could have a predisposition to any disease that actually this woman doesn't have, but are just consequence of the pregnancy. So those both cases, antibiotics and pregnancy, are also in the frequent questions of our report, are not recommended in those cases. I will explain briefly 2 examples of endogenous factors that affect the oral microbiota. I will start with the diet. A high sugars intake, we have known since always that it affects our oral health, and this is due to the microbiota. Well, this increase in this high sugar intake allows the growth of acidogenic bacteria. These bacteria are the ones who fermentate sugars, free sugars, into lactic acid. Therefore, there is an increase in the concentration of lactic acid, and this leads to a decrease on the pH. We have an acid pH in our mouth, and this is the environment where caries happen. It damages the teeth, and these bacteria are the ones who grow in the surface of our teeth when we have a caries. On the other hand, a diet rich in fiber and dairy products, it helps with a balanced microbiota. This is because it will allow the growth of bacteria that they consume the lactic acid, and they metabolize it and give as a result different metabolites that are not harmful for our teeth or for our health in general. And as they are consuming this lactic acid, they will increase again the pH to normal values, so we won't have that acid pH in our mouth that could harm our teeth. Another example will be alcohol consumption. Alcohol consumption, in the same way as sugar intake, it increases the growth of gram-positive bacteria. And among these bacteria, there are the acidogenic ones, such as Streptococcus mutans and Lactobacillus. Streptococcus mutans is the species that is most known for causing caries, and also Lactobacillus, the whole gender, is known for causing this disease too. So alcohol consumption also will increase caries risk. But not only this, but also ethanol, that is the alcohol that there is in alcoholic beverage, it's metabolized to acetaldehyde by oxidation. This process usually happens in the liver. In the liver, ethanol, it's metabolized to acetaldehyde, that is again metabolized to another not harmful molecule. Unless there is really a too high consumption of alcohol, in which case it will be saturated the process, so the acetaldehyde will accumulate. And when the acetaldehyde accumulates is when we have harmful consequences in our health, because this compound is carcinogenic. It inhibits gene reparation and even some methylation process that could affect oncogenic regions, and therefore increase the risk of cancer. As this is happening in the mouth by oral bacteria, when there is an alcohol consumption, we are generating acetaldehyde in the mouth, where there are not the enzymes that metabolize that acetaldehyde, so it will spread in the body. And this is a way in which bacteria affect the cancer risk through alcohol consumption. As a curiosity, there are also research published about the benefits of wine consumption, moderate wine consumption, red wine, in the risk of caries. It decreases this risk as it contains some organic acids that inhibits the growth of Streptococcus mutans, that, as I said just before, it's the main cause of the caries. Going into the report, and now I will explain also some of the traits that we have in the report. We will find an introduction with a really detailed explanation of the methodology, sorry, and the structure of the report in a really easy way to understand it for everyone, even without a scientific knowledge, so your clients will perfectly understand the results. Also, the most frequently asked questions, then a summary, so we can have in an overview of all the results in just 2 pages, and then the detailed results with an explanation of every trait and the main bacteria. So this is what a summary will look like. Here we will find always the profile. The profile contains if the oral microbiota of this person is similar to a smoker profile or not. And this is really important because this allows us to adapt the consecutive algorithms when we are calculating the risk. So we don't apply the same algorithm to a smoker person than to a person who doesn't smoke, and we are not biased by that, even though smoking will be always an increase of the risk. Then we have the pathologies. We have at this moment 5 traits, 3 related to oral health and 2 related to systemic health, that are lung cancer and pancreatic cancer. Then the main bacteria that will be described, and finally some bacteria that are harmful for our body. Here we have lactobacillus, that is this one that appear here in red. But we study also others, and we describe also others, but those will only appear when they are found. An example is Porphyromonas gingivalis, that I will talk about it now just in a moment. In this report, there isn't Porphyromonas gingivalis because this person didn't have Porphyromonas gingivalis. This person, this is an example actually, but here there wasn't. I mean, it's not clients, of course, but it's real data as example, and it doesn't have Porphyromonas gingivalis. But if in case it was found, it will appear here, as well as other bacteria that we also look for them as are pathogenic. We will talk about them in the report if are found, such as Neisseria gonorrhoeae, that is the causant of gonorrhea. No? So then we will have the rest of the bacteria until the 100%. And like in all our reports, there will be always a page explaining how the results are organized. So going to Porphyromonas gingivalis, this bacteria, it's really important in oral health, and it's the main pathogen of periodontitis. This is an anaerobic bacteria, strict anaerobic, so it only grows in the absence of oxygen. That already gives us a clue of how bad it could be, the fact that we have so deprived of oxygen areas in our mouth. And it's not the only cause of periodontal disease. In fact, it can be developed periodontal disease without this bacteria, but it really increased the risk. It has been really demonstrated how strong is the correlation between this bacteria and this kind of infection of periodontitis. Here we say that we consider pathogenic concentrations over 0.1%, and this is because we set there our detection limit. In case it's this bacteria will appear when it's positive. There will be always a little explanation, and like in all our reports on bibliography and reference, so you can find more information about it. Then it will appear periodontitis, and it's important the fact that these appear separate, because as I said before, you can be negative on periodontal disease, but you can have a risk of periodontitis depending on other bacteria. This infection, the periodontitis, is one of the most common in humans, and it affects around the 14% of adult population. The main causes are poor oral hygiene and smoking that allows the growing of the harmful bacteria because of the deprivation of oxygen. Then there are also, and this is really important, growing evidence that link periodontitis with systemic disease, such as atherosclerosis or Alzheimer's disease, among others. And I will talk about this later, but it's so important that it's not only related with oral health, but it has a really huge impact in our systemic health. And as I just said, we can have this without having porphyromonas gingivalis, because it also depends on other bacteria and or this dysbiosis state. For example, an increase of Prevotella and an increase of Peptostreptococcus, among others, will be affecting periodontitis at the same time as that a decrease on the anti-inflammatory bacteria will also affect. It will appear if it's unfavorable or unfavorable result, and if there is risk of this infection or not. Because here we will be talking about risk, because there is a really direct correlation between the bacteria and the periodontitis, and there is a causality that has been proved.
So as I was saying at the beginning, it's a totally breakthrough. I mean, you can really take, I mean, this test before you are getting sick on your mouth, and you know, the doctors will know how to do to avoid this disease. So it's just an amazing thing that takes us closer to our purpose. That at the end, our purpose, final purpose, is this world where you go to the doctor not when you're sick, but when we're still healthy, but you go to the doctor to not getting sick. So, and this product is just like that. It's not about predisposition. It's about a big risk that you have that is very easy to take action on these risks in order to avoid the disease. So it's just great.
Thank you, Nacho. Then I'm going to continue also still on the oral cavity and talking about oral cancers. Oral cancer is a term that is used to talk about a group of cancers affecting the oral cavity and the neck, like larynx cancer, oropharynx, tongue cancers, and all of these. I mean, altogether are the 6 most common cancer in the world. There are risk factors such as smoking and alcohol consumption, and there has been found strong associations with oral microbiota. And here I'm not going to talk about risk. Here I want to talk about associations, and I will be talking about associations because even though there has been hypothesized which change in the microbiota can lead to the oral cancer and which pathway follow to increase this risk, the scientific literature that there is now available, it doesn't talk about causality. We see associations, and we are sure that it exists, that associations, because science has proven it in this way. And however, the causality of the cancer as a consequence of the oral microbiota has not been proved. So we'll talk about associations in these cases as different from periodontitis. So as you see in this case, there is no association. But as I just said, it has been hypothesized and proved some mechanisms that could be involved in this association with cancer. One of them is chronic inflammation, and I will talk about inflammation also later, because as I guess we all here know, inflammation is one of the main risks for cancer. It doesn't matter from where comes that inflammation. A chronic inflammation will always increase the risk of cancer, and this balance in our oral microbiota will increase this inflammation, because it will increase pro-inflammatory cytokines, and it will also increase metalloprotease, and therefore we will have in all the area, in all our oral cavity, constant inflammation. Then there is also the metabolites, oncogenic metabolites, that are consequence of the metabolism of bacteria. And here we have the reactive oxygen species and also reactive nitrogen species, as well as other metabolites such as nitrosamine. These are also risk factors that has been described in the last years as a consequence, as a cause of cancer. Not cause themselves, only these metabolites, but as a risk factor. It has also been proved that the oral microbiota can alter the epithelial barrier and also can modulate the epigenome. And through modulating the epigenome, it could increase oncoepigenetic alterations, and it will increase the expression of some oncogenic oncogenic genes. Sorry.
You have to understand that we are in the very front end of science and technology in this microbiome test, oral microbiome, because as the DNA research are being done for a long period of time, there are a lot of research on DNA and diseases. Oral microbiome is quite new. So yes, there are things that are very clear. I mean, there are some bacteria that if you have it in your mouth in some quantities, you will probably going to get a disease in your mouth, right? And then we see some other associations. That's why we talk about risk, we talk about predisposition, and we talk about associations, because at the end we know for sure that there are some associations, but we still, in some cases, we don't know if it's a consequence or is or the bacteria is what is bringing the problem, right? So there's many research that are going on with different hypotheses. We are looking at them. As soon as these hypotheses are real facts, they will be in our test. So as you can see, our DNA test is developing all the time and getting bigger and bigger. We just released DNA sport test, a new release that is like 20% bigger and better than our previous version. Microbiome is going to be even more, because it is quite new. Everybody's seen that microbiome is so important for everything, but everything is being discovered now. So it will change very quickly. Thank you.
Thank you. Yeah, exactly. It's a growing field, and every day there are publications about it. So we are just there to update it as soon as possible. We have now the tools to update it, because we have all the bioinformatic pipeline already developed. So now for us, it will be easy to continue adding traits to our report as soon as the science advance. Then I want to move to systemic health, because it has been proved that the oral health is not only related to oral cavity. Oral health has an impact in our whole body. The mechanism why this happens has already been described. As I said before, inflammation. Again, the inflammation in the mouth doesn't stay in the mouth. It alters all the immune system, and it causes systemic inflammation. But not only that, but also the oral microbiota can migrate to other parts of the body. There are different microbiota in different parts of the body. This migration of the oral microbiota to these other parts can cause that the others microbiota start a dysbiosis. Starting a dysbiosis status. So in this way, it will be affecting all our systemic health. And to see an example about this, I'm going to talk a bit about lung cancer. Lung cancer is the second most common cancer globally. We know the major risk is smoking, but there is also like a 20% of the people who dies of lung cancer that has never smoked, because there are other risk factors, such some carcinogens, pollution, or lung infections. And in the last years, there is growing evidence on the role of oral microbiota in lung cancer. Here again, we don't talk about the risk, because what it has been proved now with really large research with 1,000 of participants, it's the association. Again, it's not known the causality, but this has been done with smokers and also with non-smoker participants, and it has been demonstrated that this association exists. And the papers would talk about its about the dysbiosis that can be generated in the lungs, because the lungs has always been thought as a sterile tissue, but in the last year we know that it's not like that. We have also a respiratory microbiota, and this one can be changed by the oral microbiota, because through microaspiration, this bacteria in the mouth can arrive to the lungs. As well as bacteria in the air, the same, can arrive to the lungs. But this bacteria in the mouth, if they arrive to the lung, it will cause a dysbiosis on the microbiota, and in this way, it could increase the risk of cancer. How can this happen? Through several pathways. First, the bacteria could help to angiogenesis, so to the creation of new blood vessels. And this is one of the main hallmarks of cancer, because it helps the cancerogenic cells to spread. Then it causes also, again, inflammation, and it also impaired the immune response. It can also arrive to evasion of the checkpoints of the immune response, other of the hallmarks of cancer. Then it can just cause a genotoxicity by affecting directly the DNA. And also there is some research on therapy resistance, but this is out of what our report is about. But just to point that it's really important, the effect that the oral microbiota can have even in the resistance to drugs against lung cancer.
This is what I was talking about before. This is totally new. I mean, there is no other lab in the world that is able to see associations with disease in the lungs and diseases in pancreas. It's a total breakthrough. We are the first company, the only company, and this, I think, is going to change a lot the way the physicians takes decisions about these diseases. So we're really proud of this part.
Now the last slide, just to finish this, I anticipate some before about the association that there is between periodontitis and systemic disease. And in this case, I want to talk about Alzheimer's disease, as I think it's one of the main disease that population nowadays is worried about, and it's also the main cause of dementia. And as the longevity increase, the number of cases continue increasing, and we all want to prevent this disease. And there is a lot of research on understanding the pathogenesis behind it. No? And well, here this trait is not in our report. We don't talk about Alzheimer's directly in our report, but that is because still it hasn't been found a direct association between the oral microbiota and the Alzheimer's disease. The research that has been published are not conclusive, so we cannot talk about that. We are not going to use them, as the conclusions are not really clear yet. But there is a lot going on in that field.
Yeah, that's where we're talking about, that this is going very fast. And right now, yes, there is an association between periodontitis and Alzheimer, and there is a real association between oral microbiome and periodontitis. Now, it is in between, so there is not yet a real association between the microbiome and Alzheimer. It's indirect, but research are going on. So what today is indirect, that is good enough to take care about it. Maybe in the future there is a direct link. We've seen of all these different relationships between our DNA, external factors, our microbiome, and more, all this relationship has been really discovered and well documented. I mean, the scientific world is putting all together and start to understand what's going on in our body. So, a few years ago, nobody was talking about microbiome. And a few decades ago, almost nobody talked about DNA. Now we know DNA. Now we know the microbiome is important, and more we are discovering what's the real chemical path one factor and the disease. So it's going to be amazing how many things we're going to discover in the next 10 years, and we'll put it on the table as soon as we have it.
Exactly. And just to mention the mechanism that can be influencing this link between periodontal disease and Alzheimer's disease. I talked before about it, inflammation. This chronic inflammation and systemic inflammation could be also affecting the brain. And in the last year, there is a lot of research about neuroinflammation and its involvement in neurodegenerative disease. So this seems to be the main pathway, while periodontal disease is affecting the neurons and therefore Alzheimer's disease. There is also proof that in dead bodies of people who died with Alzheimer, it has been found bacteria related to periodontal disease, such as Porphyromonas gingivalis, the one that we talked before, and also Fusobacterium nucleatum. However, it's not clear how these bacteria can pass through the barrier in the brain, the hematoencephalic barrier, but it has been demonstrated in this brain. There is also here a dual relationship, because also people with Alzheimer and dementia in general tend to have a poorer oral health, therefore they have more risk of periodontal disease. But what we think that there is the real importance, and what we want to point out, is that people with periodontal disease are in a higher risk of early Alzheimer development in the early age. So I think this it's really important to have in mind when we talk about taking care of the oral microbiota and of the oral health in general, as it's not only affecting the oral health, but the systemic health. So that's why I wanted to finish with this, and I just want to thank you all for being with us this afternoon and for your attention to this webinar.
Thank you so much, Débora. This has been so helpful, even for me. We are going to go for the questions for some minutes. If there's any question we don't have time to answer, don't worry, we will send you the answer in private, as we have your emails. So the first one, it's about the difference in using microbiome or microbiota terms. So is the same, or are any difference?
Yeah, actually are used as synonyms in most of the cases, and out of the scientific context, they can be used as synonyms. In fact, a lot of people are looking for a microbiome test, but what they are actually looking for is for a microbiota test. But to explain shortly the difference, the microbiota test, it's based on the taxonomic classification. So the bacteria and the relative abundance of these bacteria that we have. And on the other hand, the microbiome test will involve the specific variants of these bacteria. That's not what we are doing here, even though, yeah, most of the tests, for not saying all of the tests that are in the market that they talk about microbiome, are talking actually about microbiota, because we talk about taxonomic classification always. But yeah, that out of a scientific context could be used as synonym words.
Okay, thank you so much. And the next question say, should there be minimum or maximum amounts of bacteria in absolute numbers? Does dysbiosis occur when bacteria exceed a certain amount?
No, there aren't absolute numbers. The numbers that we use are media with its standard deviation, because it changes a lot between different individuals. No, therefore we cannot talk about a value that we set and we say if you are over this value or under this value, you will be in risk of anything. Dysbiosis, it's actually related with disequilibrium. So we have a decrease of a kind of bacteria and we have an increase of other kind of bacteria, and this is causing a disequilibrium, because for the kind of metabolism that these bacteria have, we know that the metabolites that they will be producing will be harmful for us.
Okay, thank you. And the next question says, I saw in the example on the website that one result was positive, but it was in red color, and I was puzzled. Is giving positive negative? I think I can answer this one, but basically in the traffic light scale we use at 24Genetics, we use the red color for saying this is unfavorable for your health. So this is not a good result. But Débora and Nacho, if you want to add anything else to this answer.
That's perfect.
Okay. So the next one says, if the report says I am positive for periodontitis, for example, does that mean I have periodontitis or that I am at risk for it? And can the dentist do anything to prevent me for developing it if I don't have it yet?
No, it's we always talk about risk. This is not a diagnosis test. But anyway, here, as we mentioned, it's a really high risk. Like if we are telling you that you have this system that it could be green, orange, and red. If it's red, you are in a moderate risk, but if it's orange, sorry, you're in a moderate risk, but if it's red, the probabilities are really high because you are in a really high risk. But this has to be always confirmed by a dentist. And yes, a dentist can do something to prevent it. In this way, we have the usual oral care that the dentist will do with or without this microbiome test. But if a dentist is on microbiota, based on the results of this report, he or she will be able to apply a specific prevention or specific treatments for the periodontitis. So instead of giving you a general drug or doing you a general treatment, he or she could specialize. No, like we're going like to the precision medicine also in oral health.
Okay, thank you. And the next one says, what does association mean? I have not quite understood why in some case you speak of association and of risks in others. This is a good one.
So just to sum up, risk, we talk about risk when there is a causality demonstrated. So if it has been demonstrated that a bacteria cause a disease, there we will be talking about risk. We talk about association when we don't know if the bacteria is a cause of the disease or it's a consequence of the disease. Therefore, we cannot tell anyone that they are at risk of disease if it has not been proved that the direction of the causality. In these cases, we will be talking about associations. Hope it's more clear now.
I think so. Thank you. And the next question say, we all know that sugar affects teeth, but is its detrimental effect on teeth produced through the microbiota, or does it have any direct effect? Is caries always caused by unbalanced microbiota?
Yes, the caries are going to be goes always for this bacteria because the sugar itself, it doesn't damage the teeth directly, and it's not able to decrease the pH on your mouth. Therefore, these are the bacteria, that the ones that make it. There will be the possibility that this doesn't appear in the report, because caries can be caused by different kind of bacteria, and sometimes are not found in the saliva, but are only in the surface. And if the amount is small, you won't find it in the saliva. But the fact of finding in the saliva and being able to say that, for example, your caries has lactobacillus will help dentists to give the right treatment. Apart from like the classical ones, it will help them to adapt the treatment to solve it. So yeah, the damage to the teeth is through the microbiota, and therefore caries is caused by this unbalance of the microbiota.
And in the bacteria results, you see a blue bar, which I understand is one that marks the normal parameters. What are those parameters based on, or on amounts of bacteria?
These are relative amounts. We don't talk about the absolute amount of the bacteria, because that is not actually what is important for health. Is a relative amount in percentage. These what we talk about, the normal or the common amounts of bacteria, are based on the Human Microbiome Project. That is the biggest project of microbiome that has ever been done. It's oral microbiota, another kind of microbiota, and they conduct really interesting research. And they also publish all this information about what is the media of people of different microorganisms in saliva. But here, as I think I mentioned before, having a higher or a lower bacteria than the media doesn't have to mean that you have a health problem. That will depend on the rest of bacteria that are found in your mouth.
Okay, thank you again. And we're going to read the last question, that is, what is the difference between main bacteria and other bacteria?
The main bacteria are the ones that are most frequently found in the saliva. We chose this one based on the Human Microbiome Project again. The ones that are most frequently found are the ones that we thought that will be more interesting for our clients. There are also in the main bacteria that are not so frequent, but that are the most important for oral and systemic health. So we decided to include them for that. And then in the rest of bacteria, there are all the bacteria that we found in your mouth.
Okay, thank you so much. So I think this is those has been all the questions we have about the microbiome test. Some other people is asking about the business partner program, so I will answer those questions at the end. I don't know if you, Nacho or Débora, want to say something else before I go on with the partner program options.
Well, I mean, I want to talk about how to put this product in the market, and it's mostly about how to put our DNA products in the market. Look, DNA is affecting our health and wellness. I mean, yeah, I think right now everybody agrees on that. So what you will be able to see in the market is that there are some doctors that they don't care about DNA, some hospitals they don't care about DNA, some hospitals they care about DNA. Even with the skincare clinics and even sports trainers, some of them they care about DNA, some of them they don't care about DNA. All right, what's the point on this? At the end, when you are selling any kind of product, doesn't matter if you are a sport trainer or a physician or you are a hospital. Okay, sometimes you can work in the old ways because your customers don't have idea about the technology that is going on, right? So maybe you are in a hospital and you don't use pharmacogenomics, and maybe you are a dentist and you don't care about oral microbiome. But you know, if some other competitor, maybe a hospital that is on the other side of the street, start using pharmacogenomics, and another dentist is using again pharmacogenomics, the customers, you know, once they prove the technology, once they have the pharmacogenomics or they have the oral microbiome test in their hands, will never understand that, you know, a dentist do not use this thing, and they will not understand that the dentist is caring about you to not get sick instead of just getting cash when you get sick. So it is the same for DNA than for this product. You guys have to explain to the customers that I'm not talking about the final customer. I'm talking about the clinics and dentists and hospitals. You have to tell them that they can be the last one taking the good things that technologies bring it to the customers, or the first one bringing the solutions to the customers. And probably their business in a long term is going to be impacted pretty much about that. So at the end, if you are the last dentist taking this technology to the final customer, you are going to be in a very bad position. But if you are the first one taking this technology to the customers, you are going to be in a very good position, because the people is going to talk about you. You know, our purpose is to speed up the use of genetics, and we will be able to do that pushing hospitals and stuff, or showing our product to the end customers. What we're trying to do is to do both, but first we love to show the product to the end customers, because, you know, if this product, nobody knows about this kind of products, nobody will demand it. So well, it is not the same because we are not the same company, but Steve Jobs, when he took the iPhone to the market, he didn't go to any shop or any big distributor, phone distributor. He took the iPhone to the final customer. And when you do that and your product is really good, your first customers are going to be what is called in marketing the early adopters. Those are the ones that, you know, the kind of guys that they want to have the very last technology, and when they love it, they talk about it. So those are the clients we need to look for. We don't want maybe the biggest customer. If we are talking about dentists or odontologists, we don't want to hit maybe the biggest one. We want to hit the opinion leader. We want to hit that one that is always at the front end of the technology. And same thing with final customers. We want to talk to these guys that are the first one buying the last iPhone or the last Tesla or whatever is on the market that is making a huge difference. Those are the ones that have to buy our products, but because they're gonna love it, and they are going to be the best selling team, because they are going to talk all the time about that. And if we talk about dentists or clinics, we have to look for those ones that, you know, they are very well motivated on having the best solution, the best technology for their customers. Once we get that, we get those ones, the rest will follow. You know, they will get a lot of advantage with this kind of product. They will grow a lot on their sales, and the rest will follow. And we have to remember that this is a tool that is good for everyone. It is good for our distributors like you, it's good for the physician, dentist, and staff, and it's good to the final customer. To the final customer, because they will not have a problem in their mouth, because it's going to be solved before they have it. For the dentist, because instead of, you know, fixing one teeth and making one shot low revenue action, they will be able to sell something that they have not been selling before. That is a DNA test, in this case a microbiome test, that is something that is not cheap, is making good revenue, and it's additional revenue because they have never done it before. And at the same time, they're getting a lot of loyalty from the customers because they're showing that they are in the very front end of the technology and they're bringing the biggest value they can give to the final customer. And you can repeat those tests. I mean, DNA test, you take it only once, but microbiome test, you can take it once in a while. So it's something that you make revenue today, but maybe in 6 months, but maybe in 1 year and stuff. And also because our DNA test and microbiome test, they're growing all the time. When you have a customer today, and maybe you don't talk to him in 1 year, after 1 year, we have a new update and you can knock again on his door. So it's great for loyalty. It's great for showing value to a customer that is not even giving you revenue today, but you have something that you can give him for free. That is an excuse to start talking again and to sell the next product. So good for the final customer, good for the dentists or clinicians and everything, good for our distribution and business partners because they can sell again and again and knock at the doors of the customers maybe in 1 year time or 2 years time. You can offer right now the sports our sports upgrade, and you can offer that for your customers in some cases even for free. So that's a good thing for you guys, and of course for the overall DNA testing industries is just a great opportunity. Absolutely. So that's the key thing. Go for the early adopters, go for those ones that are willing to test the very last technology that is useful for the people, and the rest of the players will follow. Thanks.
Thank you so much, Nacho. I think this has been so insightful for all the prospect partners that are here today with us. So thank you. And I just want to take the last minutes for going and showing you again our business partner program. For those who already are not a business partner, but are thinking about it and also asked about, do you have OEM conditions? How about me selling through other companies? I just want to remind you that here in our business partner program website, you can find this table where you're going to find all the options for partnership solutions. And if none of those ones work or you think it's not going to work, please do not hesitate to ask us, because we are happy to have a Zoom call or just a regular call to discuss it with you and try to find together the best way to collaborate. So I just want you to go to our business partner program again, or if you already saw it, just send us an email at sales@24genetics.com or partners@24genetics.com, and we will be happy to have a chat with you and find together the best solution. And this is everything for today. We're going to have more webinars for partners presenting the new products, also some others dedicated to commercials plan and how to help you help you increase your sales. So I hope to see you again soon. I want to give Nacho and Débora, I don't know if you want to say goodbye.
Yes, thank you. Yes, thanks for joining this webinar. And I think one of the questions is that if we are going to have intestinal microbiota test, yes, we are going to have that, and some other microbiome test. As you know, the pipeline we have built is ready for any kind of microbiome, and yes, we're already testing some collection kits, and as soon as we have it, we'll let you know. Yes, we will develop all our new products that are helping the people to take best decisions about their health and wellness, and everything that we can read through genetics, we'll have it in the future. Microbiomes are going to be the very first ones. And on our website, you have already all the information about our microbiome test. You can even download an example in our website. It is the same kit, it is the same saliva. If you are selling an all-in-one, you don't have to give another kit to the customer. So the same kit that you are selling our test, we don't need anymore. We take some saliva for the DNA test, some saliva, we send it to another country where we do the reading, and we will be able to, with the same tube of saliva, make both things. Yeah, be ready for a lot of new products. You will have it, and they're going to be just awesome for you guys. Thank you.
Okay, so bye for now, everyone.
Bye. Thank you very much.
Thank you.